So in this video, I'm going to go over to you guys what glycolysis is, show you all the different steps involved in it, and also explain to you guys how it gets regulated by the cell. So how the cell controls how fast glycolysis goes. So glycolysis at its most basic form is going to be the pathway by which glucose, which is a monosaccharide, a simple sugar, gets converted into pyruvate. And pyruvate is a really important starting compound when it comes to making a bunch of different amino acids, a bunch of different other compounds like oxaloacetate and acetyl coenzyme A, which actually goes through something called the Krebs cycle to make more ATP. So it's really important that our cells are able to synthesize pyruvate, and glycolysis is the primary mechanism by which our cells are able to make it. So glycolysis is also a catabolic process because we are breaking down glucose. We're not synthesizing it, we're breaking down glucose to make pyruvate. Its products are also more oxidized. That means the pyruvate is going to be more oxidized than glucose. Glycolysis also yields 2 ATP net and 2 NADH and H+. So NADH, H+, and also something else called NAD+, are something called electron carriers. What they do is they basically shuttle electrons between different biochemical reactions. We're going to see those, in a bunch of different chemical pathways. So it's good to, get used to it. So we're going to make 2 ATP, 2 NADH, and H+. There's also going to be 2 different phases to glycolysis. There's something called the energy investment phase, where we're going to put in 2 ATP. We're going to invest 2 ATP because we're going to yield 4 ATP in our energy yield phase. It's also called the energy payoff phase sometimes. And energy investment is going to be phases 1 through 5. Energy yield is going to be phases 6 through 10. And I'm going to just move this up a bit. There we go. So this is going to be our first step of glycolysis. I'm going to write that down. First step. And what we're doing is we're taking glucose, which is our starting compound, and we're cleaving, we're not cleaving, but we're taking off this H at the end of our OH group there, and we're tacking on a phosphate group. And this phosphate group is coming from our ATP. So this is in our energy investment phase, so we're putting in 1 ATP here. And ATP, as you know, is adenosine triphosphate. I'm going to write out ATP like this. That's going to be our adenosine group, and then these are going to be our 3 phosphates. What we're doing is we're hydrolyzing this last phosphate there, and we're tacking it on to our 6th carbon. And we know it's our 6th carbon because we can, count and check it out. So that's 1, 2, 3, 4, 5, and 6. And that's why it's called glucose 6-phosphate, because we're taking off that hydrogen group there, and we're adding on a phosphate group to the 6th carbon. So this is going to be our second step of glycolysis. What we're doing is we're taking the glucose 6-phosphate that we made, and we're converting it into fructose 6-phosphate. Now, glucose and fructose are both monosaccharides. They're both simple sugars, and they're isomers of each other. So they have the exact same molecular formula, but they have different structures. You can see that glucose has 5 carbons in its main ring there, and 1 carbon at the end. Now, fructose is going to be pretty much the same thing. We're going to have 4 carbons in the middle. We're going to have 1 carbon there and the 2nd carbon over there. But it's still going to retain that phosphate group on the 6th carbon there. Besides that, it's going to be the same compound. And the enzyme that does this is called phosphoglucose isomerase. For step 1, I forgot to go over it, but the enzyme that does that, it's called hexokinase. So this is going to be our third step of glycolysis. What we're doing is we're taking our fructose 6-phosphate that we just made, and we're tacking on a 2nd phosphate group to the 2nd side. So the enzyme that does this is called phosphofructokinase 1, and it's important to keep in mind that there's a very similar enzyme called phosphofructokinase 2, PFK2. And the difference between these 2 enzymes is that phosphofructokinase 1 is going to add a phosphate group to the 1st carbon in the chain. So if we count again, that's going to be 1, 2, 3, 4, 5, 6. phosphofructokinase 1 is going to add a phosphate group to the first carbon in our chain, and phosphofructokinase 2, PFK2, is going to add a phosphate to the second carbon in our chain. And phosphofructokinase 2 is actually involved in regulating the activity of PFK1, and we're going to look at that later on in the video. And what we're doing here is we're priming this compound to get cleaved in the middle. Our next step is going to involve breaking this apart in half, and we're going to get two compounds with one phosphate each. So these are going to be steps 4 and 5 of glycolysis. So step 4 is where we're taking this F16BP that we made, fructose 1,6-bisphosphate, and we're breaking it up into two, like I said before. So we're going to have two compounds that are produced in pretty much equal amounts. We're going to have DHAP, which stands for dihydroxyacetone phosphate, and we're going to have GAP, glyceraldehyde 3-phosphate. Now the difference between these two compounds structurally might look different, pretty similar, but the thing is only GAP can go through glycolysis. Only GAP can go to step 6. So between these two, we're going to need a mechanism, a method to convert any of the DHAP that gets produced by our aldolase. We need to convert that into GAP so that all of the products can move on with glycolysis. And that's going to be our step 5. We're going to have an enzyme there called triose phosphate isomerase, and its entire job is to convert between DHAP and GAP. Now, it does seem that our step 5 is not reversible, but it's an equilibrium reaction, right? So some GAP is still going to get converted to DHAP, and it's going to proceed in both directions. But in general, this reaction is going to be favored in favor of producing more GAP, because this GAP is going to get depleted by future reactions. As this GAP gets used up in step 6 to make the next compound, according to Le Chatelier's principle, DHAP is going to be depleted. We're going to use DHAP to make more and more GAP, and that's going to drive this reaction further. So this is going to be step 6 of glycolysis. What we're doing is we're going to take all of the GAP that we made, both from our aldolase and from our triose phosphate isomerase, and we're going to oxidize it. So we're going to convert it from GAP into, I forgot to write down the compound. It's 1,3-bisphosphoglycerate, or 1,3-BPG. So yeah, we're going to oxidize the GAP to make 1,3-BPG, 1,3-bisphosphoglycerate, and we're going to use up one of our electron carriers. We're going to use up NAD plus and a phosphate group, and we're going to reduce it to NADH and H plus. And this phosphate group is what gets tacked on to our other side there. And the enzyme that does this is called GAP dehydrogenase, or GAP dehydrogenase, and it's also called GAPDH sometimes. It's just an abbreviation. You're going to see this a lot because GAPDH is often used as a housekeeping gene. As a housekeeping gene for like northern blots and other molecular biology tools. But in glycolysis, we just know it as an enzyme that oxidizes our GAP there. So this is going to be our 7th step of glycolysis. What we're doing is we're taking our 1,3-BPG, or 1,3-bisphosphoglycerate, and we're removing one of these phosphate groups, and we're adding it onto an ADP. So it's kind of the opposite of what we saw with our hexokinase mechanism, or not mechanism, but the hexokinase step. ADP is adenosine diphosphate, so it's 2 phosphates instead of 3. What we're doing is we're taking off that phosphate group there, and we're adding it on to our ADP to make ATP. And we're producing a different compound called 3-phosphoglycerate, and the enzyme that does this is called phosphoglycerate kinase. Now, a confusing thing about this enzyme is that when you see the name of it, phosphoglycerate kinase, you might think, oh, it's a kinase. It's a protein that adds a phosphate group to our phosphoglycerate, but it actually does the opposite. It's going to act as a phosphatase on our substrate, and it's going to add on a phosphate group to our ADP to make ATP. Now this is step 8 of glycolysis. What we're doing is we're taking our 3-phosphoglycerate and we're yanking off that phosphate group, and we're adding a different phosphate group to our oxygen there. Now, this isn't an isomerase, this is a mutase, because in an isomerase reaction, the group that gets yanked off is the exact same group that gets added on to the other location there. But in a mutase, this phosphate group is taken off, but it's not the exact same phosphate that gets added on. There's a different phosphate that comes from the environment that gets added on there. And you might see it in the mechanism of this reaction, which we're not going to go over, but it's just important to keep in mind that this isn't an isomerase, it's a mutase. So this is our 9th step of glycolysis. I've just made a small fix here. This oxygen used to be on the main carbon chain, but it's supposed to be branched off. So I fixed that. But anyways, we're going from 2-phosphoglycerate into something called phosphoenol pyruvate. And it's also sometimes called PEP. And the enzyme that does this is called an enolase, and we're also releasing a hydrogen or a water in the process. So this is going to be our last step of glycolysis. What we're doing is we're taking off that phosphate group and we're adding it onto an ATP, just like we did before with our phosphoglycerate kinase, and we're forming our last ATP for glycolysis. And we're also forming pyruvate, which, as we talked before, has a lot of different fates that can that it can go through. And the enzyme that does this is called pyruvate kinase. Pyruvate kinase might sound like it's a kinase that acts on the pyruvate. You might think that it's a kinase that's going to add a phosphate group to its substrate, but no, it's taking off a phosphate from the PEP and it's adding it onto an ADP. So those are the only 10 steps of glycolysis. Now I'm going to talk about how they get regulated. So here I've written out all the major steps in glycolysis that are involved in regulation. So you can see that I've skipped a bunch of steps there. I've only written out steps 1, 3, and 10 because they're the only ones that are involved in regulation of glycolysis, and we're just going to discuss them in detail now. So the first step, the hexokinase step, is important because G6P acts as an inhibitor of hexokinase. So as we perform glycolysis more and more, and we get more and more G6P, it's going to signal to the cell that we're already doing it a lot. So we don't need to continue going on and producing more and more pyruvate when we already have a bunch of glucoses that are already going through glycolysis. So it's going to act as a negative feedback loop to inhibit the activity of hexokinase, to slow it down. And also, this step is important because glucose normally can enter and leave the cell through something called facilitated diffusion. So glucose is large enough that it can't just diffuse through the plasma membrane. But if this is our cell, glucose has these things called GLUT transporters, GLUT transporters, in the cell wall, or in the cell membrane, that allow it to leave and enter the cell freely through facilitated diffusion. But if you add a charge to the glucose, if it becomes glucose 6-phosphate, now it can't just use an ion channel, not an ion channel. It can't just use the GLUT transporters to enter and leave the cell. So it kind of locks it in. It locks in that glucose molecule for glycolysis, for other metabolic processes. So that's also a pretty important part of this step. Step 3 is pretty much the most important step when it comes to regulation of glycolysis. PFK1 is the first committed step of glycolysis. After you make F16BP, that molecule has to go through glycolysis. It can't stop. Before that, glucose can just get stored. G6P can get stored as glycogen, so it doesn't have to go through glycolysis. F6P can go back because step 2 is a reversible reaction. So F6P can go back to G6P and it can get stored as glycogen. So it doesn't have to go through glycolysis either. But after you make F16BP, it has to go all the way through and make pyruvate. So that's the first committed step. So there's a lot of incentive for the cell to go and influence this step a lot. So there's a bunch of different mechanisms that are used by the cell to regulate the activity of PFK1. The first one is the levels of ATP, ADP, and AMP. So PFK1 has 2 sites for ATP binding. PFK1, ATP binding sites. That's the first one. And it has a catalytic site and a regulatory site. When ATP binds to the catalytic site, it's going to lower the activity of PFK1. So this ATP is what's used up by the PFK1 to make F16BP. It's the ATP that actually gets hydrolyzed to come up with a second phosphate to add on. So if we already have ATP in the cell, if we already have high levels of ATP, if we're already in a high energy state, there's no incentive for the cell to make more and more ATP. This cell is better off storing the glucose for later on, for scenarios when the cell does need more energy. So ATP binding in the catalytic site is going to lower PFK1 activity. And the other thing, the opposite, is going to be high levels of AMP and sometimes ADP. AMP and ADP sometimes are able to bind to a different regulatory site of PFK1. And when AMP and ADP bind to that site, that means we're in a low energy state. AMP is adenosine monophosphate. So where adenosine triphosphate had 3 phosphate groups attached, adenosine monophosphate only has 1, and adenosine diphosphate only has 2. So that means we don't have a lot of ATP. We're in a low energy state. So that's going to signal the cell to make more and more of F16BP. It's going to make more and more of this committed molecule, and it's going to ensure that this goes through glycolysis, and we're making more ATP and more pyruvate. So that's going to enhance the activity of PFK1. The second factor is pH. Low pH is going to lower the activity of PFK1, and that's relevant because this pyruvate can get converted into something called acetyl coenzyme A, which goes through something called the citric acid cycle or the Krebs cycle. And the citric acid cycle is all about harvesting electrons from an acid. It starts out with citrate or citric acid, and it yanks off a bunch of electrons with each step, and that's what the Krebs cycle is about. But what's relevant for glycolysis regulation is that citrate and all the other steps are acids, are acidic. So when we have a low pH, we already have a ton of pyruvate. We already have a ton of citric acid. We're already making lots of ATP. We don't need any more, right? So again, in this scenario, the cell is better off storing the glucose and using it later on. So this is also going to downregulate the activity of PFK1. The third, and I'd say most complicated way to regulate PFK1 is through something called F26BP. We had F16BP before. Now we have F26BP. Now F26BP is produced by that PFK2 enzyme that we talked about. And the production of this is actually a little complicated. So F6P, fructose 6-phosphate, can be used to make F26BP, the same way PFK1 uses F6P to make F16BP in step 3 of glycolysis. F26BP is the exact same as this, but instead of it being in the first position, it's going to be in the second position. That's all it is, but it plays a completely different role, right? It's going to act as an allosteric activator of PFK1, and it's made by PFK2, but only in certain scenarios. PFK2 is a bifunctional enzyme. PFK2 can act as a kinase, which is what it's doing here, as a kinase, or it can act as a phosphatase. It can act as a phosphatase, which means it's going to remove F26BP. So when PFK2 is acting as a kinase, it's going to make more and more of F26BP, which means we're going to make more glycolysis. We're going to, do more and more of glycolysis there. PFK1. Yeah. And if we have more PFK1, we're going to increase the level of glycolysis. But PFK2, when phosphorylated, it can act as a phosphatase. It's going to do the opposite. It's going to take all this F26BP and it's going to convert it back to F6P, fructose 6-phosphate. If it does that, that means that we're going to have less and less of F26BP, which is going to be our allosteric activator. So if we have less F26BP, we're going to have less PFK1. We're going to do glycolysis less. So it's going to have the complete opposite effect. So you can see how PFK2 can get pretty complicated, right? So if we have regular PFK2, it's going to act as a kinase. It's going to promote glycolysis. It's going to make more of this activator. And if it's a phosphatase, if it's phosphorylated, it's going to do the opposite. It's going to take this F26BP. It's going to convert it into F6P. So as we deplete this activator, we're going to have less PFK1. We're going to have this glycolysis, and it's going to bring the whole thing down a notch. And the way that PFK2 decides, or it doesn't decide, but the way that PFK2 gets switched on and off between these two states, between the phosphorylated and the unphosphorylated state, is because of the activity of a couple of enzymes. So PFK2's kinase activity is activated by an enzyme called insulin, which you might have heard of. And insulin uses something called the AMP kinase pathway. I'm pretty sure, I'll put in a note if it's incorrect, but I'm pretty sure it's the AMP kinase pathway, which is going to activate its kinase activity. It's going to promote its kinase activity. And glucagon, which is the opposite enzyme of insulin, glucagon is going to lower its kinase activity. Glucagon is actually going to help phosphorylate the PFK2, and it's going to promote the phosphatase activity of PFK2. And I'm pretty sure glucagon uses the PKA pathway. I'm also going to check this later on and correct it in the comments or in the video if it's incorrect. But yeah, you can see how these two hormones are involved in the control of PFK2, which controls the activity of, which controls the amount of F26BP, which again is an allosteric activator. Remember what allosteric means, right? If this is our enzyme, if this is PFK1, that's our active site. That's where our F6P binds. We're going to have an allosteric site on the other side. That's where F26P binds. And allosteric means it doesn't have to compete with the enzyme, with the substrate for binding. It can just go into this other side and bind and influence the activity of PFK how it wants. It doesn't need to compete with F6P. So again, recapping, PFK2 can either be a kinase or a phosphatase. PFK can either make more F26BP and promote glycolysis, or it can be a phosphatase and make less F26BP, convert it back into F6P, and inhibit glycolysis and lower glycolysis. And the activity of that, determining whether it's a kinase or a phosphatase, is determined by the activities and the signaling involved with insulin and glucagon. And we're going to see these two pop up again in our last step in pyruvate kinase. So pyruvate kinase has 4 major isoforms, and actually that needs to be ADP, ATP. All 4 major isoforms are controlled by F16BP. F16BP is the compound we made before. F16BP, its presence is going to activate, it's going to promote the activity of pyruvate kinase. And that's because if we've already made a lot of F16BP, we need to ensure that we have enough pyruvate kinase and that it's working properly to make more pyruvate. It can't just get stuck in the middle, right? So F16BP is going to be an activator of pyruvate kinase. And out of those 4 isoforms, specifically in the liver isoform, which is only one of 4, it's a rare isoform, but in the liver, PK also has a bit more complexity to it. So when PK in the liver, again, is phosphorylated, it's going to lower its activity. And when PK is not phosphorylated, when it's just PK, it's going to work better. It's going to work more. And this phosphorylation state is, again, controlled by insulin to promote it and glucagon to lower it. Again, that's only in the liver isoform. That's one of 4 major isoforms of this enzyme. All of them are controlled by F16BP, but only the liver isoform is controlled by insulin and glucagon. And yeah, that's glycolysis. Again, we have 10 major steps involved. It's a catabolic pathway. Its products are more oxidized. We're going to make 2 ATP net and 2 NADH at H+. We have 2 major phases. We have our energy investment phase, which is, let me draw that out. That's our energy investment phase. And step 6 through 10, which is right to left here, is going to be our payoff phase. In the investment phase, we're going to put in 2 ATP, 1 in our hexokinase step, and 1 in our phosphofructokinase step, because that's where we add in a phosphate and a second phosphate. And then in our yield phase, we're going to make 4 ATP. So we're going to net 2 ATP. And yeah, that's glycolysis.